Good afternoon, everybody. Today we'll start with lesson number 5, Origin and Evolution of Life. Students, this year due to COVID-19, 25% syllabus has been reduced in all the subjects. In this lesson number 5, we will be studying only 2 articles, but it is only for this year students, okay? 5.6 and 5.13. See, 5.6 article is Modern Synthetic Theory of Evolution. The modern synthetic theory of evolution describes the evolution in terms of genetic variation in a population that leads to the formation of new species. It is the result of true synthesis of all biological disciplines, that they are genetical, ecological, anatomical, etc. Besides these things, importance was also given to mutations and natural selection. The main contributors of this modern theory of evolution were Fisher, Haldane, Mendel, Morgan, Stebbins, etc. There were many. The theory describes the evolution of life in terms of genetic variation in a population that leads to the formation of new species. The theory also describes the genetic population or Mendelian population. The theory also describes the genetic population or Mendelian population, gene pool, and the gene frequency. It explains the contribution of factors like genetic variation, reproductive and geographical isolation, and natural selection. Now see here, students, when there are group of people or group of individuals, they form one population. These populations are actually the small groups, okay? And when they are in the population, the process of interbreeding takes place. So this is what is Mendelian population, okay? So an interbreeding group of organisms that share a common gene pool is referred as Mendelian population. Now what is gene pool then? Gene pool is the total number of or sum total of genes of all individuals in a population. Any changes in the gene pool affects the population. Obviously, changes within a gene pool occurring from generation to generation is called microevolution. Allele frequencies in a population may change due to gene flow, genetic drift, natural selection, and mutation. Now what is gene frequency then? Gene frequency, it is the frequency of occurrence or proportions of different alleles of a particular gene in a given population. Next slide. Now see here, students, to study the concepts of modern synthetic theory, how this theory was put forth to 3 categories. One is genetic variation. Instead of category, I think we can use the word factors or concepts. 3 concepts are put forth. One is genetic variation, second one is natural selection, and third is isolation. Gene mutation. Now genetic variation, why genetic variation is taking place? It is because of the following characters, that is gene mutation, genetic recombination, gene clone, genetic drift, chromosomal aberrations, which are deletion, duplication, inversion, and translocation. Now let's take up one by one. For genetic variation, it is the difference in DNA among individuals. What is genetic variation? It is the difference in DNA among individuals. There are multiple sources of genetic variation, including, and what are the sources? They include mutation, genetic recombination, etc. Okay, so let's take up the first one now, gene mutation. It is a permanent alteration in the DNA sequence that makes up a gene. This leads to the change in the phenotype of the organism, causing variation. When mutation occurs within a gene, it is called point mutation or gene mutation. Second is genetic recombination. It is the exchange of genetic material between different organisms, which leads to production of offspring with combination of traits. Recombination, it just means that something is being combined. Okay, that is already it is present and again it is being recombined. That differ, combinations of traits that differ from those found in either parents. Recombination in the form of crossing over. Recombination can take place because of crossing over, which occurs between the homologous chromosomes in order to increase genetic diversity by changing the assortment of the genes. Now, let's switch on to the gene flow now. What is gene flow? It is also called gene migration. Flow, the word moves from one to another. Okay, or it transfers from one to another. That is the gene flow. Gene flow is the transfer of genetic material from one population to another. It can take place between two populations of the same species through migration and is mediated by reproduction and vertical gene transfer. Students, what is this vertical gene transfer? Now, that means from parent to the offspring. Gene flow also alters gene frequency, causing evolutionary changes. Now, next is genetic drift. It is a mechanism of evolution in which allele frequencies of a population change over generations due to chance, that is sampling error. Genetic drift occurs in all populations of non-infinite size, but it affects are strongest in small population. That means what? It can occur in all the population types, in all the different populations, small or bigger, but it is strongly, it affects the small population. Drift increases the amount of genetic differentiation among populations if no gene flow occurs among them. It has significant longer-term evolutionary consequences. Sudden immigration or emigration of individuals in a population changes gene frequency in the resulting population. So this was about the genetic drift. Now let's come down to chromosomal aberrations. Chromosomal aberrations, these are the alterations in the structure of individual chromosomes, or chromosomal aberration may occur spontaneously or by induction. So what is aberration? These are the changes, okay, and they can occur either spontaneously or they can be induced also, that is by induction. Such changes may result in quantitative alteration of genes or rearrangement of genes. The breakage and reunion of chromatic segments result in a number of abnormalities in the chromosome structure. While we were studying the chromosome structure, that time also I told you this. Thus, origin of structural changes is caused by breaks in the chromosome. Okay, so this is all I am talking about the chromosomal aberrations. Now we will see that how many types, 4 different types of such aberrations are there. One is deletion, duplication, inversion, and translocation. Deletion or deficiency. It is also known as deficiency type. Okay, so part of the chromosome will be lost or it will be deleted. Okay, deletion or deficiency, both are same. It means that either the part of the chromosome will be lost or it will be deleted. Okay? So depending upon the deficiency or deletion, it is on two different types. One is known as the terminal deficiency or deletion, and other one is known as intercalary. So what is terminal? Terminal means at one end. Okay, so when part of the chromosome from at one end is removed, or it is cut, or it is deleted. So that is terminal deletion. It is a single break. I'll tell you in the statement form. It is a single break near the end of a chromosome would be expected to result in a terminal deficiency. Then second type is the intercalary. Intercalary, the word inter means between something, between two. Okay, so here the chromosome will break at two places, right? So two breaks occur in a chromosome. A section may be deleted, it may be deleted, and an intercalary deficiency is created. I hope you understand this, what is deletion and deficiency. Next comes the duplication. Now, what is the meaning of duplication? The double. Okay, so part of chromosome will be either added it will be added, and therefore it gets duplicated. Okay. Then third one is inversion. Now what is inversion? That is inverted. Okay, upside down. Now a chromosome is there, right? A part of it is removed and it is again attached to the same place but in an inverted position. Okay, so let's see. Sections of chromosome detached and reunited in reverse order. So there is no loss or no gain of the genes over here. Okay, in this, the broken segment gets reattached to the same chromosome in an inverted position. Last is translocation. Translocation, parts of chromosome detached, right, and joined to non-homologous chromosome. This is also known as transposition. Here, I hope you remember jumping genes, students. These jumping genes, they are the DNA sequences that move from one location on the genome to another. Okay, and that is known as the transposons. Right. So, what is translocation? It is a part of chromosomes which is detached and joined to the non-homologous chromosome. That is transposition takes place. It is naturally affected by the jumping genes, that is transposons, present in the cell. And I told you what is this jumping genes? They are nothing but the DNA sequences which can move from one location to the another in the genome, right? So this was about the translocation, that is chromosomal aberration. Now let's go to the next one, that is natural selection. Now, natural selection, the best example given is industrial melanism. First of all, let's understand what is this natural selection. Students, the meaning of the word, that is natural, itself means by nature, selection, of course, by nature selects. Okay? So, it is the process through which populations of living organisms adapt and change. Individuals in a population are naturally variable. That is, they are all different in some ways. It means that some individuals have traits better suited to the environment than the others. So in this case, the heritable traits increase an organism's chances of survival and reproduction, which results in the evolution of organisms. So what does it mean? That an organism is there, the nature, its surroundings or its habitat has been slightly changed, it could adapt with that and then it continues one generation to the another. So an evolution of organism takes place. Okay? The fittest will be at the selective advantage, obviously. Best example here is given of industrial melanism. Students, in Great Britain, just before industrialization, there was a moth called Western Butylaria. Western Butylaria. Okay, this moth was having two different colors. One was white wing and other one was black wing. Okay, so trees in this area, trees in this Great Britain, near the industrial area, they were having, on their trunks were present the lichens, because of which the lichens color, because of the lichens color, this white wing could mimic with that. Okay, and we could see that white wings were more when compared to the black wings. This all was before industrialization. Okay, because when birds came to peck them, to eat this, pick up these moths, they could see only the black winged ones, because white wings were camouflaging with the lichens. So what happened then? It is just in a story form, students, right? Industrial melanism, the darkness of the skin, feathers or fur acquired by a population of animals living in an industrial region where the environment is soot darkened. Soot matlab jo धुआं आता है, that smoke that comes. Okay, it is the appearance of dark melanic forms of some organisms like moths in the industrial region, where the heavy emission of carbon from the burning coal has colored the background. Now the evolution of the peppered moth, this Western Butylaria, call it as peppered moth, is an evolutionary instance of directional color change in the moth population. As a consequence of air pollution, during the Industrial Revolution. Now, in Great Britain, before industrialization, I told you white-winged moths, that is Distal butilalia, were more in number, and black-winged moths, that is Distal carbonaria, were comparatively less in number. Moths being nocturnal, active during night. Nocturnal means active during night, used to rest on tree trunks, which were covered with a lichens. White-winged moths camouflaged with lichens and escaped from being victims of the predatory birds. On the other hand, what happened? Black-winged moths, while resting on tree trunks, were easily noticed, becoming victims of the predatory birds, getting reduced in the number. Now, during Industrial Revolution now, what happened? Black sooty smoke was released, covered and killed the lichens on the tree. Okay, that is trunk turned black due to pollution. Trunk of what? Trunk of tree, right? Advantage to black winged moth now, because the trunk has turned black due to the soot, and they were having black wings, so they were able to camouflage now with the tree trunk. So number of black wings increased. Now what happens to the white-winged moths? They became victims of predatory birds. Therefore, their number reduced. And therefore, this is known as natural selection. Okay, I hope you have understood this. Often it is asked in the exam. You can be asked for the short notes also, industrial melanism, or give what is natural selection, explain industrial melanism. Now the third part is the isolation. Now this isolation is what, students? Separation. Okay. So isolation can be of two different types. One, because of the geographical conditions, we call it as geographical isolation. That is, the populations, they are separated by means of because of the river, because of the mountain, etc. Okay. And other can be the reproductive. Now isolation, separation of the population of a particular species into smaller units, which prevents interbreeding between them. Now barriers preventing gene flow or exchange of genes between isolated populations is isolating mechanism. Now here see type of isolating mechanism. There are two different types. One is pre-mating or pre-zygotic we call it, and other is post-mating or post-zygotic. Yeah? Now here you see habitat in pre-mating, it can be because of the habitat or ecological conditions. It can be seasonal or temporal. It can be ecological. It can be mechanical. Right? And under post-mating, we can see gamete mortality. It can be because of zygote mortality, or it can be because of hybrid sterility. Students, you can just see here, hybrid is only written, sterility has been shifted over here. I'm sorry, try to understand this. Gamete mortality, zygote mortality, and hybrid sterility. Okay, now I'll tell you one by one. Two types, I said geographical isolation. We'll take up first this geographical isolation. It is the physical separation of two populations by the geographical barriers. Barriers can be mountain, it can be river, or it can be large areas of climatic differences. The separated populations develop distinct gene pool. For example, Galapagos finches. Population of finches found on the different islands of Galapagos evolved separately and therefore split into 15 different species. In some it is given reference 26 also, approximately. So I use the word approximately. They all look different and utilize different food sources. Students, it is simple. It is just like that. We all are human beings, but we are staying in a different states, different areas, different places in India itself, isn't it? And we can compare, when two individuals, one from south and one from north, they come in front of us, we can distinguish very clearly that he is a South Indian or he or she is a North Indian. So same thing over here also. So this was because of the geographical isolation. Okay. And next is the reproductive isolation. Separation of a single population into several subunits. What is the reproductive isolation? It is the separation of a single population into several subunits brought about by the changes in genetic material, changes in the gene pool, and changes in the structure of genital organs. And this is therefore known as reproductive isolation. Now, two types of reproductive isolations. These are, I have shown you here, pre-mating and post-mating one. Pre-mating, also called pre-zygotic isolating mechanism. The members of the population are isolated by habitat, that is ecological. Okay? Seasons of maturity, and this is temporal, seasonal or temporal I have given there. Okay, so seasons of maturity will be different. Then that means the difference in the sexual maturity. Then ethological means different mating behaviors will be there, and mechanical means difference in structure of reproductive organs. Thus, interbreeding is avoided. So I hope you have understood that what is the pre-mating problems kya kya hai. One is habitat, the place where they live, seasonal, different breeding seasons, ecological, different mating behaviors will be there, and mechanical, that means the structures, the reproductive structures, or the organs, they are itself different. Okay? Next, you see post-mating or post-zygotic. In post-mating or post-zygotic isolating mechanism, it will include gamete mortality. Gamete is formed after the fertilization. Okay. So I'm sorry, I'm extremely sorry. Gamete is formed after the reduction division, right? Male gamete and the female gamete. So chances are that due to limited lifespan, if union of the two gametes does not occur in due time period, the gametes will die. The gametes will, there will be a mortality. This is known as gamete mortality. That means what? The egg is ready and the sperm, the male gamete is ready and the female gamete is ready, right? But they're not, they have got a particular lifespan, and if they do not meet in, they do not fertilize within that, or if there is no fusion between them in that particular time lifespan or the time period, then obviously they are of no use, and this is known as gamete mortality. Then next is zygote mortality. After egg is fertilized, forming zygote. Zygote may not develop due to some reason in zygote is, that is known as the zygote mortality. That means fertilization has taken place, but they are not, it is not developing further. Okay, so this is also known as the, that is known as the zygote mortality. Next is the hybrid sterility. That means gametes are formed, zygote fertilization has taken place, zygote was formed, development was there, the individual, the hybrid developed everything, but it is not able to participate in the process of reproduction. This is known as hybrid sterility. That means inhibition or suppression of the reproductive capacity of F1 hybrid. That is, hybrid becomes sterile due to failure of proper gametogenesis. Okay, so this was about the everything about the theory, synthetic theory. Next, we will see the human evolution. That is the next article 5.13. That is human evolution. Now, in human evolution, students, we will first see the introductory part of it. The study of human history is known as anthropology. Man belongs to an order, primates, and primate includes apes, monkeys, lemurs. Lemurs means they look cat-like, okay, and man. Then anthropoid apes include monkeys, apes, and man. Apes and man are devoid of tail. Apes include chimpanzees, gorillas. See, I have given you the slide in a stepwise manner. Okay, apes include chimpanzee, gorilla, orangutan, and gibbon. They are the nearest relatives of man. So these all, they are the nearest relatives of man. All of these belong to superfamily, that is hominoid. Okay, hominoid, and man belongs to family Hominidae. Whenever it comes a, that means it is a family. Okay, like Malvaceae, Taplioniaceae. In botany we have studied all this thing. So same way here, it is Hominidae. Now the characters of Hominidae. They had erect posture. Obviously, erect posture means there will be bipedal locomotion will be there. They walk on the two legs. Then they were slightly S-shaped. They had slightly S-shaped vertebral column. Then their teeth, it was crescent shaped. Arrangement with reduced canines, expanded cranium, reduced brow ridges. Sorry. Forelimbs were slender and thumb was opposable. So these are the characters. Besides, human evolution Now, these characters are there. Then human evolution is the evolutionary process that led to the emergence of anatomically modern humans, beginning with the evolutionary history of primates, in particular genus Homo, and the genus it is Homo, and leading to the emergence of Homo sapiens as a distinct species of the hominid family, the grey apes. Now here you can see the flow chart over here. The first one is the Dryopithecus, which is on the last line. See, you can see the arrow mark, apes, Ramapithecus, Australopithecus, Homo erectus, early Homo sapiens, Neanderthal man, Cro-Magnon man, and Homo sapiens sapiens. So we all belong to this Homo sapiens sapiens. Next you see, same evolutionary history of man. Evolutionary history of man was traced with the help of fossil remains. So how did we come to know about that? The evolution of history of this man's evolution, human beings evolution, or so it was because of the fossil remains. And the first one, as I said, it is Dryopithecus. They are the common ancestors of apes and man. It is more ape-like. And second is Ramapithecus, it looks more man-like. Then Australopithecus, they are the African ape man. Homo habilis, they are the handy man. These are the other names of that. Homo erectus, these are two different types were found. One was Homo erectus and found in Java, so Java man, their name was, and Homo erectus pekinensis, they were found in the fossil was found in the Peking of the China, this is Peking of the China, and therefore it is known as Peking man. And then Neanderthal man considered to be the first civilized man. Why they were considered to be the first civilized man? Actually, they buried the dead ones. Homo sapiens, again of two different types. Homo sapiens fossilis, they are referred as the Cro-Magnon, and Homo sapiens sapiens, they are referred as the modern man, which we all belong to. Now next you see, we'll take up the characteristics quickly of each this type. Dryopithecus, they had common ancestor of ape and man. Fossils from Bilaspur, that is Himachal Pradesh. Hairy body, more ape-like they were. They had brow ridge accent, and their brow ridge was absent. Teeth arrangement was U-shaped, semi-erect posture, forerunner of gorillas and chimpanzees, and they existed about 15 million years ago. Students, in the book, if it is written 15 MYA, so that MYA is millions years ago. Next is Ramapithecus, existed about 15 million years ago. It is more man-like. Fossils from Shivalik Hills, walked erect on its hind feet, had small canines like man, ate hard nuts and seeds. Their jaws were there, and their dentition, it was V-shaped. Then Australopithecus, which is also known as the African ape man. In East Africa, fossils was discovered by Ramond Dart. It is the connecting link between ape and man, and erect posture with 4 feet height, bipedal locomotion. Whenever erect posture, understand that it's with a bipedal locomotion. It had a lumbar curve. Dentition was just like man. Hands used for non-locomotory functions. And used for non-locomotory functions means what? If it is 4 legs, then it will be it will walk on 4 legs, isn't it? But here it is bipedal, right? So just compare that. Hands used for non-locomotory functions, and then use stone weapons. They use stone weapons, and why did they use weapons? Obviously, they must be hunters, so they use some weapons, right? Cranial capacity was 600 cc, cubic centimeter. Brow ridges projected over eyes. Their chin were absent. They ate fruits. There were of 2 different types, Australopithecus robustus and Australopithecus boycei. Next is Homo habilis or handyman. Fossils were discovered by Leakey. It is considered to be the first human. Cranial capacity was 650 cc. Dentition more like man, smaller molars. It was erect. It did not eat meat. It did not eat meat. This is a unique character of it. Made tools from stones. They had primitive shelters and probably could communicate also. They were considered to use fire. That means they used fire. Then Homo erectus. They were found 1.5 million years ago. Their cranial capacity 900 cc. They ate meat, they were omnivorous, and they used fire also. Their height was 5 feet. Prognathous face, that means the jaw was projected forward. Bony eyebrow ridges, white fleshy nose. Homo erectus means the upright man. Okay? There are two types. One is Homo erectus erectus. It was found in Java, hence called Java man. Then Homo erectus picensis, found in Peking in China, hence called Peking man. Its cranial capacity was 1075, that is 1075 cc. Next is Neanderthal man. It was found across Europe and Central Asia. It used hides to protect body, short stature, erect, strong mandible, and cranial capacity was 1400 cc. They constructed flint. Now, what is this flint, student? It is a hard grey rock which consists of pure silica. Okay, so they constructed flint tools. They buried the dead, and it is a first civilized man. Next, Homo sapiens. They evolved in Africa and then moved to the rest of the world. Now here, understand the things properly. They evolved in Africa and then moved to the rest of the world. And they were found 10 to 70,000 years ago. Two types, Homo sapiens fossils. They are also known as the Cro-Magnon. They were tall and erect, and their cranial capacity was 1660 cc. They were cave dwellers. They lived in caves, carnivores, and used sophisticated tools. Now, the second type were Homo sapiens sapiens. They are referred as the modern man. They were erect. They are erect. They domesticated animals. They started agriculture around 10,000 years ago, and their cranial capacity was 1450 cc. Now, these Homo sapiens sapiens students, they were divided into four ethnic groups, right? So, ethnic means the race-related groups, like we say Negroes, Russians, and like okay. So, Negroids. First one was Negroids. These Negroids were seen in Africa. Next was Caucasians. They were found in England and Italy. Next was Eastern type. The third type were Eastern type, and Eastern type were found in Japan, China, Australia, and India. Then Mongolites, the people living in Mongolia. So these are the four different ethnic groups in which these Homo sapiens sapiens they were classified. Okay? This is all I wanted to tell you today. I stop here and will continue with the next in the next session. Thank you so much.